Hello, thank you for the opportunity to speak today. So this was a collaboration project between Professor Benny Chain, who you may have heard of, who he developed a bulk TCR sequencing protocol, and between Professor Tessa Crompton, who she's a sort of T cell development specialist and does hedgehog signaling as well. So this project was a sort of collaboration between using, quite an interdisciplinary collaboration of using bulk TCR sequencing to look into T cell development. And the work is published as a reviewed preprint here in eLife. And this was my PhD and postdoc project. And it was to look and determine the differences between the fetal and the young adult TCR repertoire. So luckily already we've had a little bit of a recap about T cell development. So I'll go through a little bit quickly. But as said before, hematopoietic precursors, they enter the thymus from the bone marrow. And you have like the double negative stages, which are CD4 and CD8 negative. And quite importantly, at the double negative, from about the double negative to double negative 4 stages, you get TCRβ rearrangement and the expression with the pre-TCR. And then later, and you get a proliferation stage. Later you get maturation into double positive, which is the CD4 and CD8 expression. And at this stage you get TCRα rearrangement. And then you get the expression of the TCRα-β chain. And then you will get CD4 or CD8 commitment in either SP4 or SP8. And positive and negative selection. So negative selection if the TCR signals are either too strong or too low. So just to kind of have a little recap about the fetus and fetal thymic T cell development. So unlike the adult, the precursors come from the fetal liver in the fetus. And they generally emigrate into the thymus at around embryonic day 10 to 12. So if you think of the lifespan, of the sort of gestation of a mouse, it goes from embryonic day 0 to around 20 or 19, depending on the strain. So at around E10 to E12, you get hematopoietic precursors seeding the thymus. And then from around E11, you get like a kind of proliferation in the first double negative cells. And around E14, you get a larger population of double negative cells. And at E16 is when you start detecting the first double positives and the first immature single positives or CD8 positive ISPs. And at E18 is when you actually do start detecting some mature single positives at this stage. And just after that, you get birth. So the major differences between the fetal and the young adult T cells. So as I already said, in the fetus, everything comes from the fetal liver, which is quite different environment. Then in the young adult, that will come from the bone marrow, and that will come from the bone marrow for the rest of the life of the mouse. And in the fetus, the thymus is still forming, so those structures are still forming. So that's a different microenvironment to the young adult, where the thymus will now be developed. And a huge difference is the absence of the enzyme TDT, which adds the N nucleotides and the junctional diversity. So this is not present in the fetus. While in the young adult, just after birth, there's the expression of TDT, and therefore there'll be the addition of N nucleotides, so there should be increased junctional diversity. And also there have been several studies, and a few studies in particular, that have shown that, for example, neonatal CD8 positive T cells can become short-lived effectors after infection, while if an adult is taken, those would become long-lived memory cells. So there is a functional difference between these T cells that are deriving from the fetus versus the adult. So the design of experiments in this paper, I looked just before birth, so I wanted to look just before the addition of TDT. So did embryo stage E18.5, so there will be some single positives at this stage. And then we looked at the young adult as well, so 4 weeks old. And this should have already have a developed thymus and already have homeostasis in the thymus. Then we isolated the thymocytes, and we looked at the double positive and the CD4 and CD8 single positives. We then did a bulk TCR sequencing protocol, which was developed by Benny Chain, where you extract RNA and then you tag each mRNA molecule with the unique molecular identifier, the UMI. That way it can then be error corrected for PCR bias later in analysis. This is using his decombinator pipeline. You can then error correct for the PCR bias, And then you can look at other sort of downstream analysis and in R. So first we just wanted to look and clarify the clonality of the fetal repertoires and see if this confirmed any sort of previous work. So we looked here is on the x-axis, you can see there's the TCR abundance. So this is the number of how much each clone type, how many copies of each clone type there is. And then it's plotted against the proportion of the TCR repertoire in a log-log plot. And as you can see, there is an increased clonality here in the fetal, which is in red, as you can see here. So there are sort of more expanded clones, and we fitted this to a power law exponent because power law is known to fit quite well to this type of distribution. And as you can, and we looked at this is across each of the replicates of each individual mice, and as you can see, the clonality was generally increased in the fetus versus the young adult. So the young adult had already what we think is a more diverse repertoire. Then I wanted to kind of confirm this, so I looked at just expanded TCRs. So we ranked all of the TCR sequences by their abundance and did analysis on just the top 1 percentile, here. And as you can see here, the proportion of the repertoire that is represented by expanded TCRs is more in the fetus in red here than it is in the young adult in these two populations, in the double positive and the CD4s. And then I also just looked roughly like sort of at the TCR abundances that were above that particular threshold. And again, in the CD4, there was like an increase in sequences. So this is all leading to what I sort of looked here to sort of confirm. So looking at TCR diversity in the Shannon, there was an increase as we expected in the 4 weeks in the double positive and in the single positive population. So the fetal diversity, TCR diversity, is really reduced in the beta chain. And then we wanted to look at the variable and the joining gene usage and see if this varied in fetus versus young adult. And quite interestingly, when we look at heat maps of the proportional gene segment usage, so we have TRAV, TRAJ, TRBV, and TCRβ-J. As you can see in all of the heat maps, they cluster by stage predominantly. So the embryo of the fetus clusters on its own in comparison to the young adult. So they are really using different V and J in comparison to one another. And very interestingly, we noted that there was a bias towards the use of the 3' TRAV here, and an increase in the 5' TRAJ segments in the fetal repertoires. And also in adult repertoires, the CD3 negative low double positive, so these are cells that should have not gone through positive selection at this stage, while CD3 positive double positive are the ones that should have gone through, already going through some positive selection. They actually cluster separately in these heat maps. So that's sort of suggesting the effect of positive selection on the V and the J gene usage. So then I did some principal component analysis, and I looked at the V times the J combinations. And just similarly to the heat maps, as you can see, they cluster by PC1. By age. So here you have the fetus and here you have the young adult. And the young adult is quite sort of clustering quite tightly together, while the fetus are a little bit more dispersed, so they experience a little bit more variability in the V times the J. And they did not really fully segregate by cell type, while here there's a little bit of a clustering by cell type in the young adult. So this suggested to us that MHC restriction and positive selection could have a weaker impact on the TCR repertoire in the fetus compared to the young adult. So then we wanted to look at this and do some heat maps to actually compare, so the same results of the PCA, but now actually looking at heat maps to look at the usage. So this is proportional gene usage in red. So when it's red, it's increased in the fetus compared to the young adult, and when it's blue, it's decreased in the fetus compared to the young adult. And as you can see here, quite strikingly, the fetal double positive repertoire favored these combinations that were from the 3' location of the TRAV and the 5' location of the TRAJ here. And this bias is lost in the single positive populations. So this suggests that this is perhaps the effect of sort of positive selection, or the selection process is evening out the differences. And then to sort of look again and sort of confirm what we kind of already thought, is to look at the shorter CD3 length and sort of compare the CD3 length of the young adult and the fetus. So as we expected, the double positive, in all the populations, the fetus had a shorter mean CDR3 length and also had a shorter mean non-template insertion length as well. So this is sort of, again, confirming that TDT is adding those extra nucleotides and that the fetus is more closely encoded by the genomic sequence. So then we also wanted to look at sharing between the repertoires and confirm. So we did the Jacquard index, and in this particular case, we compared like to like. So we compared the fetal repertoires with the other fetal repertoires and the young adult to the other young adult. And we looked at the shared whole CD3 putative amino acid sequences. So these are predicted productive. And as you can see here, the fetus actually has an increased sharing in comparison to the 4 weeks, especially at the double positive stage, but also at the single positive stage as well. So then, in order to kind of investigate this further, I then wanted to look at sort of sequential amino acid triplets. So I wanted to divide up the CDR3 sequence into k-mers. And so as you can see here, you sort of divide it up into all the possible sequential amino acid triplets. And quite interestingly, this showed the reverse result. So it showed decreased sharing in the fetus compared to the young adult in the double positives and the CD8s. So we were thinking this could be the result of the selection of recurrent motifs by contact with the peptide MHC during positive selection in the young adult. So even though in the young adult there will be sort of more addition of nucleotides, so that could explain why in the young adult there'll be less sharing. However, when you actually divide up the sequence, there's still some recurrent motifs that are being selected. So then we want to look at the CDR1 versus the CDR2 usage. So they've clustered really nicely by the CDR1 versus CDR2 usage, as you can see here. And they cluster generally by life stage, but also by cell type, with again the fetus being a little bit more dispersed. And so this is kind of really showing the effect of selection on the repertoire. And then again, these are some heat maps of the same data. So we're sort of highlighting some that were clearly sort of MHC restricted in both the fetus and the young adult. So there are still some V and Js that were being MHC restricted in both, as they would only appear, some would only be after selection would appear in the CD4s, while some after selection would be more in the CD8s. But still, interestingly, the heat maps still cluster by life stage first. So this fetus versus young adult repertoires are truly quite distinct. And the double positives were positioned between the CD4 and CD8. So this is kind of, again, sort of an example of MHC restriction following positive selection. So then to investigate it further, we were thinking that a lot of this, that the usage of certain TRAV and TRAJ segments by chromosomal location could potentially be the fact that in the TCRα, there is rearrangement can keep happening at the double positive stage, so there's sequential successive rearrangements. So we were thinking that potentially this could be to do with the differentiation of fetal thymocytes versus adult thymocytes. So in the fetus, it's generally a large synchronized wave of cells, whereas in the adult, it's unsynchronized, it's slower, there's homeostasis, and there's an accumulation of cell types over time. So we wanted to sort of see if this would affect the young adult population. So what we did was that we treated the young adults with hydrocortisone treatment, and hydrocortisone treatment depletes the thymocytes of double positive cells. So by day 2, there should be generally very few double positive cells. And then we waited until day 6 to then get the replenished populations. And we FACS-sorted CD3 negative low double positive and CD3 positive high double positive. So again, this is the pre-selection repertoire and sort of a more post-selection repertoire that's already going through positive selection. So yes, so we wanted to see if in the fetus progressive rounds of TCRα rearrangement are less common than in the young adult, perhaps of this result, the difference of the kinetics. So then we sort of put together the HC treated, as sort of labeled here, together with the fetal and the control non-treated. And as you can see here in these heat maps of the variable joining gene usage, the fetus clusters together with the hydrocortisone treated repertoires separately and actually has quite a distinct usage of both variable and joining in comparison to the untreated controls. So, and they actually, it's favoring by location, so they favored the 3' TRAV segments and the 5' TRAJ, just like the fetus. And so this is suggesting that this hydrocortisone treatment and synchronizing these cells actually sort of makes the young adult usage more fetal-like. And then we did again looking at the V times the J combinations, as you can see here. So this is, in pink, you have increased in the 4 weeks hydrocortisone treated, and then in green you have that it's decreased in the 4 weeks hydrocortisone treated in comparison to sort of the untreated young adult. And as you can see here, there is an increase in the 4 weeks hydrocortisone treated on this, again, this side of the heat map. So in the 3' location of TRAV and the 5' location of TRAJ, just like in the fetus. And the same, obviously, concomitantly on the same side, a decrease. So again, this is suggesting that this treatment is making the usage of the V times the J more fetal-like. And this suggests that the kinetics of the transition to the double positive cell are quite important in determining what gene segments are being used. So we think that potentially in the fetus, the cells could be going through like perhaps quite fast because they are trying to fill a space. It's a synchronized wave. While in the young adult, it is more of a homeostatic. It's perhaps a slower process. So when you treat them the same, you get a kind of more similar result in the variable times adjoining gene usage. So the main differences that we found in conclusion were that between the fetal and the young adult, there's a suggestion that the fetal thymus is producing alpha-beta T cells, properties and functions that are quite distinct from the young adult T cells, and they are perhaps more innate-like. And that the fetal TCR repertoire appears to be less governed, so strictly by MHC restriction in comparison to the young adult, with a preference for very particular gene segments, so quite a distinct phenotype in comparison to the adult, less diverse, with more clonotypic expansions, and more closely encoded by genomic sequence. So it'd be really interesting in the future to look into like the function of these cells and investigate this further. So yeah, so I'd just like to acknowledge my the lab at ICH, and my supervisor, Tessa, and Benny Chain, who were invaluable during this project. Great, and again, you can see the reviewed preprint at eLife, and also like to thank UCL Genomics and UCL Gosh as well, and my program Lido as well for supporting this project.
Really nice talk. Thank you so much. We're looking at questions now, and I'm going to ask you a question myself. And that is, when you're doing this comparison, we know that in fetal life, there are waves of different kinds of innate type cells, and it seems your data are really backing that up in terms of the restricted diversity and all. So what I wanted to know was, with the hydrocortisone-treated animals, are they getting more innate type cells because they're TCR and so forth? And is there a way you could look at that, for instance, in these, more innate type like populations that are, and would they then be, going into their respective places that they go, even though it's a much older mouse?
Yes. No, that would be really interesting. We haven't actually looked at that. We just looked more, when we've done hydrocortisone, we've more looked at just like quite a T cell lab. But yes, that would be really, really interesting to measure and look at those populations as well, because I would imagine hydrocortisone is quite a sort of broad treatment. It kills a lot of different cells. So I would imagine that could perhaps like set the balance. Perhaps there would be more or less innate cells as well. So yes, it's great suggestions. Thank you.
Yeah, it's just very interesting. Anyway, okay, so questions. Anonymous. It's the same person over and over again. Anonymous. God, they just push really hard. Okay, in the PCA for gene usage and stage, what is the % variance explained by PC1 and 2?
I can't remember off the top of my head, but there were quite a lot of principal components, so it wasn't a huge percentage of variance. So I think it was something like 40% or something like that, but I can't. And each one was different, so I can't remember off the top of my head.
All righty. And Bjorn Peters is asking, do you see differences in mate TRA chain gene usage compared to other TR alpha in any of your studies, specifically usage in embryonic versus adult?
So yes, we didn't actually look at this, and yes, it would be very interesting to look at this, especially after seeing these talks and sort of getting ideas. Yeah, that would be very interesting.
All right, okay. Is the higher sharedness of fetal CDR3s due to their shorter lengths, or does this hold when comparing only CDR3s of the same length too?
I think, yes, so I think I did do this, and it was of some particular lengths. I think, yes, overall, of the sharing. No, sorry, I didn't actually look at them individually and compare, so I can't really say. But I would suspect that, yes, some of the increased sharing is due to the fact that there are shorter lengths.
Okay. Did you happen to look at non-productive rearrangement events and see any patterns there also?
Yes, so we didn't put this in the paper because I think it's quite hard to interpret what the non-productive sequences are doing at this stage. But yes, there are patterns in the non-productive also, and they also do cluster by stage. So it does seem like there really are some also differences in the rearrangement choice of different V and J, because you're seeing that in the non-productive sequences that are obviously not being selected. So yes.
Okay, and could you scroll that up a little, please? Is that possible? There we go. Okay, sorry. Did you see differences in the CD4+CD8 composition? Thank you.
I'm not sure in what sense in terms of the number of cells. There are, it's still roughly similar that there's going to be more CD4, but the fetus does have differences, yes, but I didn't compare directly like CD4 versus CD8, no.
Okay. Were there differences in CDR3 length between the groups?
Yes. I think I'm not sure exactly which groups, but yes, there was a sort of decrease in CDR3 length in the fetus.
In the fetus?
Yes.
Okay. So, sorry, what do you, I can't hear you. Is it a puberty? Okay. Is there a puberty during gestation in humans? Is there in mice? What does that mean? How have you seen any differences between males and females?
Between males and females, we are actually going to be looking at this because this is one of our comments from all reviewers, but generally, no, I don't really see that many differences, but I will be like putting that in the revised paper. Okay, and so, can we do more questions? Oh, okay. Yeah, I don't know why you were doing that. Okay, in liver-derived versus bone marrow-derived T cells, are there differences in exonuclease activity as there is for TDT? Yeah, I'm not sure about this, but I'm not sure. I don't know.
Okay. How are the expanded fetal clones distributed in 4 young adult mice? 4 young adult mice. What does 4 mean?
I'm not sure what.
Maybe young adult mice? Maybe they mean young?
Yeah, you can see there is obviously variability in the fetus, there's more variability in the clonotypic sort of expansion, but it was significant when comparing just like power law when it's fitted. So,
fascinating data about the fetal diversity as opposed to the restriction in young adults. This seems to follow the hourglass model of development.
yes.
Okay, she's shaking her head, yes, okay. So that good point.
Good point, thank you.
Yeah. Were the same amount of cells sorted for the different subsets? Were the data sets equalized, downsized, resampled for the diversity index?
Yes, so the answer is no, we did not sort same numbers. We sorted just the whole population, but we did downsample to the smaller sample because, yes, as said previously,
to make them comparable.
It's going to change, the effect.
Okay. Beautiful. Okay, is there a change in lineage proportions, gamma delta versus alpha beta TCR?
We didn't look at gamma deltas, but yes, I know that gamma deltas in the fetus do have distinct functions. They go out, they're actually the first T cells to emigrate from the thymus, and they colonize tissue specific, and they actually remain throughout your life, these fetal gamma deltas. So yes, that would be really interesting to look at in the future as well.
And their selection is different, right?
Yes,
Totally.
their selection. Yeah. And the gamma delta selection, so the delta is related to the alpha. So we were thinking some of the changes could be potentially relating to that, the fact that the delta is nestled in to the alpha, and apparently the delta gets spliced out.
That's right.
And so then you'll get like some pairs of the alpha being chosen. And we think potentially that this is happening less in the fetus because you're getting specific ones being chosen on those ends rather than sort of dispersed on the other end as well.
Okay. And what is triggering the unsynchronized T cell development in adult mice?
Just homeostasis. So like in the fetus, there's this real push, in any developing animal to sort of get the cells, fill the space, and at that point, those single positive cells are probably quite different to a young adult single positive cell. So in the young adult, in the CD4 and CD8, there's more homeostasis. There's cells that remain there, potentially that don't exit the thymus. And actually in aged mice, even more complicated, there's cells that re-enter the thymus. So that's kind of like this thing of homeostasis, as in the cells are talking to each other and feeding in, so. Yep. Yeah. Okay, yeah. Thank you very much.
That's it. Thank you very much. That was wonderful.